In the course of this microcycle, a growth hormone is formed that is indispensable to cellular division. This phenomenon has been observed repeatedly in vitro cultures. If, under the effects of stress, or for that matter, any biological perturbation, the blood's inhibitors are diminished to any considerable degree, the somatid's microcycle evolves into a cycle that is both different and polymorphic. We then see the appearance of the various stages of a macrocycle, 13 additional phases in all. First, we notice a bacterial form that is the normal evolution of the microcycle when the inhibitors have been diminished. This endogenous bacterial form has been observed by many researchers, Germany's von Bremer being one of the first. The double bacteria which evolves from the preceding bacterial form is often observed in blood smears. Its particularity is the ability to divide itself by scissiparity to form rods. The rod form looks like a bacterial form, except that it is longer and its cytoplasm seems empty. It should be noted that this bacterial form possesses two terminal spores. The granulated double spore bacterial form has a cytoplasm filled with granulations that begin to move. The maturation of the granulated double spore bacterial form results in a mycobacteria well known to microbiologists. The cytoplasm is also self-developing. The mycobacteria that we have just seen has developed further and formed bubble-like enclaves to which it owes its name, bubble mycobacteria. Here we see the bursting of the bubble mycobacteria and the release of the elements of its cytoplasm into the medium. The yeast-like formations that result from the bursting of the bubble mycobacteria have a diameter of 4 to 5 microns. Even at this stage, they give evidence of a centrosome. The yeast-like formations proliferate and become ascospore forms, precursors of mycelial elements. From an assay form, we can observe the formation of a thallus, in which the cytoplasm gradually takes shape to constitute the young mycelial form. It is through a conjuncture and with peristaltic movements that the young mycelial form develops a thallic cytoplasm and eventually becomes an adult mycelial form. When this mycelial element reaches full maturity, its cytoplasm is extremely active. And when it bursts, it releases an enormous quantity of new particles into the medium, each particle capable of duplicating the entire cycle. Emptied of its cytoplasm, the thallus has a fibrous appearance. Incidentally, it is this residue from the somatidian cycle that is often observed in blood smears and dismissed as an artifact of coloration. It must be underlined that these forms of mycelial characteristics do not answer to any of the criteria of fungal growth. In fact, they are not affected by massive doses of amphotericin B, fungicide, or any other antifungal substance. The preceding observations lead us to a series of postulations. 1. The somatid is endowed with polymorphism, a polymorphism controlled by blood inhibitors. 2. Growth hormones are generated during the somatidian cycle. 3. In both the animal and plant kingdoms, cellular division requires the presence of the somatid. 4. When blood inhibitors are lacking, growth hormones are allowed to increase until they threaten cellular metabolism. Five, all degenerative diseases are a consequence of this disorder. At this point in my exposition, you might well be saying, nothing you have shown us so far proves that the motile particles observed in blood plasma are really somatids. I wholeheartedly agree. We will now move on to an experiment that begins with the placing of a blood smear under the somatoscope. We can see the figured elements of the blood, and among them, bright and motile particles. Could these be chylomicrons or lipoproteins? To find out, we can raise the temperature of the somatoscope's heating stage. It is usually set at 37 degrees Celsius. First, we will increase the temperature to 54 degrees Celsius, then to 70 degrees Celsius. Immediately, some of the bright points disappear, while others become immobile. However, some of the bright and motile particles continue to spin about. What do you think they are? Let's repeat the experiment, but this time with VLDL lipoproteins. Their dimension is known, from 2,000 to 3,000 angstroms. The same phenomenon is observed, and the remaining motile particles continue their movement. Now we apply the same procedure to HDLs. We know that their dimension is from 300 to 500 angstroms. Usually, HDLs cannot be detected with optical microscopes. Under the somatoscope, they are perfectly observable. As we reach 48 degrees Celsius, the same phenomenon is produced, and the motile particles are just as agitated. Even at 70 degrees Celsius. We can make two conclusions from this series of experiments. One, the somatoscope allows us to observe HDLs, and therefore particles of 300 to 500 angstroms. Two, the particles that remain motile even after having been heated to 70 degrees Celsius cannot possibly be chylomicrons or lipoproteins. Therefore, they are in reality somatids, since somatids are not at all sensitive to heat. This demonstration confirms that the somatoscope allows us to examine living material under conditions that rule out the use of either conventional optical microscopes or electronic microscopes. The electronic microscope has superior magnifying powers and a better resolution, but it cannot be used to observe living material. Furthermore, it is only the surface of the preparation that becomes visible. For its part, the conventional optical microscope, with its magnifying potential of 2,000 times, allows for the observation of bacteria, but not of viruses. The somatoscope allows both to be seen, and in their living state, and not only viruses, but any particles of like dimension. For example, the inclusions within the erythrocytes go all but unseen under the conventional microscope, but they are easily observed with the somatoscope. The detection of these particles in red blood corpuscles amounts to new pathological input, and an exhaustive study to determine its clinical significance is clearly indicated. Earlier, we saw that the somatid is endowed with polymorphism. We have cultured it in a nutritionally peptinated and sweetened medium and ascertained through experimentation that the macrocycle can be restrained when certain inhibitors are introduced at physiological concentrations. We can establish a correlation between the regulation of the somatidian cycle and the clinical stage. We can also extrapolate that in pathological cases, the appearance of elements of the macrocycle corresponds to a disturbance of the normal cycle. This disturbance can reach a critical point and transform itself into a severe immune deficiency, which opens the door to all opportunistic microorganisms. HIV, CMV, pneumocystic carinii pneumonia, PPLO, hepatitis B, etc. In the blood of people with AIDS, we can observe the various phases of the macrocycle within either the red blood corpuscles or the plasma. The fragmentation of the polynuclear's nuclei comes about in an entirely abnormal fashion, and the basis for such a transformation appears to be in the decay of the cytoskeletal structure. The platelets submit to a ballooning phenomenon, and their lifespans are greatly reduced. It is the same abnormality found in the blood of individuals with HIV. The presence of a significant quantity of filamented mycelial forms must also be noted. This is especially true in cases involving Kaposi sarcoma. The lymphocytes are subjected to profound modifications. First, we notice a ballooning of the cell on the side of the cytoplasm. Then, filaments appear on its opposite side. In the terminal phases of AIDS, up to 90% of the lymphocytes can be affected by this phenomenon. Some of these filaments elongate to form tubules. We can observe at this stage the transfer of material through a microvillus. This phenomenon has also been observed through the use of an electronic microscope at a magnification of 50,000 diameters by Lennart Nilsson of Stockholm's Karolinska Institute. It was reported in the October 1988 edition of Scientific American, volume 259, number 4. In conclusion, in a healthy individual, a microcycle has 3 stages: somatids, spores, and double spores. The presence of elements of the macrocycle indicates a disturbance of biological systems. In the light of the somatidian theory, it would appear that AIDS is an opportunistic illness, with characteristics not unlike Kaposi sarcoma, pneumocystic carini pneumonia, or esophageal candida. The disturbance of the somatidian cycle, whether caused by the use of drugs, multiple STDs, hepatitis B, etc., brings in its wake a marked weakening of the immune defenses, which in turn permits the implantation of HIV. The somatidian theory returns our attention to the presence of Mycoplasma incognitus, noticed by many researchers during their AIDS research. The presence of this microorganism may well point to a much more profound disturbance of biological regulators, a disturbance that leads inevitably to immune deficiency. Seen from this angle, we feel that new therapeutic possibilities have been opened to us, and the trials that we have undertaken as a result have been most encouraging. Thank you for your attention.